everyone, welcome back to The Explainer. Today, oh man, we are unpacking an absolute medical thriller. And honestly, the craziest part is that it's happening right now, literally inside your own body. We're looking at some groundbreaking research from the journal Gut, and it completely redefines how we view our organs, our genes, and the microscopic passengers we carry around every day. We're going to decode this hidden, incredibly complex communication loop happening between your liver, your genetic code, and the trillions, yes, trillions of bacteria living in your gut.
Okay, let's dive into this. What if, and just stick with me here, what if your gut bacteria could actually rewrite your genetic code? I know, it sounds like total science fiction, right? We've all been taught that our DNA is this locked vault. It's a fixed blueprint you inherit, end of story. But the research we're unpacking today reveals a massive biological plot twist. It turns out our microscopic gut residents are actually fully capable of manipulating our biological software. To really grasp why this is so absolutely crucial, we have to look at this staggering number, 32.4%. That is roughly a third of the entire global population. And that's how many people are affected by a condition called Metabolic Dysfunction-Associated Steatotic Liver Disease, or MASLD for short. This is really a silent, massive epidemic. MASLD has actually emerged as the leading chronic liver disease worldwide. It's multifactorial, meaning it's driven by a whole bunch of things like cardiometabolic risks, our diets, sure, but also, as you'll see in just a second, it's driven by that microscopic ecosystem living inside us.
So how does this silent epidemic actually progress? Well, it starts at step 1, hepatic steatosis. Essentially, this is just simple fat accumulation, specifically triglycerides, building up in your liver cells. For a lot of people, it stops there. But for a subset of patients, this simple fat triggers a really aggressive immune response. And that moves us to step 2, MASH, or Metabolic Dysfunction-Associated Steatohepatitis. This is where the real damage starts. We're talking severe liver inflammation, where cells get so damaged they physically balloon up. If this goes unchecked, it progresses to severe scarring, known as cirrhosis. And eventually, it can lead to hepatocellular carcinoma, or HCC, which is a highly deadly form of liver cancer. So figuring out how this progression happens, the stakes literally could not be higher.
Part 1, the 3 key players: genetics, epigenetics, and the microbiome.
So what exactly is driving a perfectly healthy liver down this dangerous path? To solve this, we need to bring in our 3 main suspects. It is not just about eating too much sugar or fat. The breakdown of a healthy liver is orchestrated by this super intricate interplay between your genetics, your epigenetics, and your microbiome. Let's break down those first 2 using a computer metaphor. Think of your genetics as your fixed hardware. This is your raw DNA sequence, including those tiny little variations we call single nucleotide polymorphisms, or SNPs. You're born with this hardware. You can't change it. But epigenetics? That's the flexible software. Epigenetic modifications don't change the actual DNA sequence. Instead, they act like biological light switches, turning your genes on or off. They do this through processes like DNA methylation, using enzymes called DNMTs, or by tweaking the histones that your DNA wraps around, using enzymes called HDACs.
Which brings us to player number 3, your gut microbiome. When it's healthy, it's this perfectly balanced factory. But when that balance gets thrown out of whack, say you get an overgrowth of certain bacterial groups like firmicutes and a drop in good ones like Bacteroidetes, we end up with something called gut dysbiosis. And dysbiosis is bad news. It compromises your intestinal barrier, creating what we call a leaky gut. All of a sudden, dangerous bacterial toxins like lipopolysaccharide, or LPS, can slip right through the intestinal wall, catch a ride on the portal vein, and head straight to the liver, where they trigger massive inflammation.
Part 2, bugs hacking your epigenome. The biological plot twist.
Now we are zooming all the way into the microscopic level, because here is where the story gets absolutely wild. We already knew bugs could cause inflammation, sure, but what else are they doing once their chemical signals actually reach our liver cells? Guys, they aren't just passing through, they are literally hacking the system. And this brilliantly illustrates exactly how. Take a look at the bottom left here. These are microbial metabolites, specifically short-chain fatty acids, or SCFAs, pumped out by your gut bacteria. Notice how they cross the gut barrier and literally move right inside the hepatocyte, the liver cell. Once they're in, these bacterial byproducts directly interfere with our epigenetic enzymes, those DNMTs and HDACs we talked about earlier.
I really want this quote from the research to just sink in for a second. The gut microbiota and their derived metabolites can induce epigenetic modifications. I mean, wow, this completely shifts our fundamental understanding of human biology. The bacteria in our gut are actively hacking and reprogramming our epigenetic software.
The chemicals they excrete are essentially dictating whether our liver cells decide to turn on cancer-causing genes or keep protective genes active. Let's look at a really specific, concrete example of this hacking in action. Take butyrate. It's a short-chain fatty acid produced by your good gut bacteria. When butyrate makes its way to the liver, it acts as a very powerful HDAC inhibitor. So by inhibiting these histone deacetylases, butyrate is essentially installing a protective software patch directly into your liver cells. The research shows that this specific epigenetic tweak actually stops simple hepatic steatosis in its tracks, preventing it from progressing into the really dangerous inflammation of MASH.
Part 3, Genes Shaping the Gut, a two-way biological street.
But wait, the plot thickens. We just saw how the microbiome rewrites our genetic software. But is it possible our human genes are firing back? Are our own cells telling the bacteria what to do? The answer is a resounding yes. Let's move to this macro view and see how this builds, because this biological conversation is definitely a two-way street. You can see the arrows forming a complete loop here. Yes, microbial components influence epigenetic modifications in the liver. But look over on the right. Host genetics and epigenetic alterations influence the composition of the gut microbiome itself. Your fixed human DNA is actually acting like a bouncer at a nightclub, deciding exactly which bacteria get to survive and thrive in your gut.
Let's break down the hard evidence for this. Take a look at the SQLE gene. That stands for squalene epicoccidase, which, by the way, is highly active in liver cancer. When this human host gene is overexpressed, it completely flips the gut environment upside down. It actually enriches a specific pathogenic bacteria, Vibrio fairfieldensis, and that drives massive cholesterol accumulation in the liver. Or look at the HIF2 alpha gene variant. Its expression literally starves off beneficial bacteria, completely destroying bile acid metabolism. So our biological hardware is literally shaping the very microbiome that, in turn, attacks our liver. It is a vicious, toxic feedback loop.
Part 4, the future of treatment, breaking the toxic loop. So how in the world do we escape this loop? If we are stuck in this microscopic cycle where bad genes breed bad bugs, which then turn around and activate more bad genes, what's the fix? Well, because we now understand that the gut and liver are this intimately linked, we don't just have to attack the liver directly. We can actually treat the gut to heal the liver. We're seeing amazing clinical trials using engineered bacteria designed to secrete helpful hormones. We're even seeing the use of bacteriophages. These are literal viruses engineered to act like microscopic assassins, hunting down and killing only these specific harmful bacteria that damage the liver.
So the crucial point is this: modern medicine is finally learning how to interrupt this toxic conversation. We can use FMT, fecal microbiota transplantation, to basically hit reset and replace a sick microbiome with a healthy one. We can use postbiotics, which is cool because it skips the bacteria entirely and just delivers the good metabolites, like those SCFAs, straight into the gut. And amazingly, we now have resmetirom, which is the very first FDA-approved drug for MASH. It works as a thyroid hormone receptor beta, or THR-beta agonist. And guess what? Emerging research suggests that even this liver-targeted drug might ultimately be working by modulating this exact same gut epigenetic axis we've been talking about. We've seen today that the boundary between us and them doesn't really exist. Your liver health is totally, completely intertwined with trillions of microbes that are actively coding your epigenome right now. But I want to leave you with a provocative question to chew on. If the bugs in our gut can successfully hack our liver cells and rewrite our genetic software to either cause or cure disease, what else is our microbiome secretly controlling in our bodies? Think about that for a bit. Thanks so much for joining me for this explainer, and keep exploring.